Hello there, so today we're going to talk about plant morphogenesis. What is plant morphogenesis? Plant morphogenesis is the process leading to and determining the form and structures of organs, which involves several overlapping developmental processes that contribute to plant morphogenesis. Among this process including growth, cell differentiation, and cell-to-cell communication. Precursor cells differentiate into specific cell types, which are organized themselves to eventually form the specialized and complex tissues and organ systems that make up the functional plant. Morphogenesis may be concerned with the whole plant, plant part, or subcomponent of a structure. Plant morphogenesis is therefore simply defined as the origin and developmental of plant's physical form and external structure. Plant morphogenesis is influenced by several different types of factors involving homeotic genes, its polarity, its positional information, and environmental factors. Homeotic genes is a group of genes that control the pattern of body formation in organisms. Plants begin life as a tiny seeds with difference in genes in expression, producing all different types of individuals. These genes encode proteins called transcription factors that direct cells to form various parts of the plant. Two classes of homeotic genes: regulatory genes that control the activity of genes and effector genes that are directly involved in a developmental process. A great deal of information is required to establish polarity and symmetry, then patterns of cells, tissue, and organs. During growth and development, various parts of the plant send chemical message to other parts such that development is coordinated. Chemical substance, nutrient or hormones from surrounding cells indicate the cell's position on the plant. Consider the differentiation of gut cells that must occur in pairs. A pattern establishment mechanism in some cells inform each cell of its location or position relative to other cells. This way, one gut cells develop to form a pore facing the wrong direction. Chemical secretion and compression or stretching are important in establishing patterns of cells and tissue. For example, in one plant, the mother cells produce a peptide and protein inhibiting other cells, but if inhibition fails, then the proper pattern does not develop. Almost all plant communication is done by transport of hormones. Hormones are organic chemicals produced in one part of a plant and transport to other part, where they initiate a response. Auxin, cytokinin, gibberellin, abscisic acid, and ethylene are the most well-known hormones stimulate plant morphogenesis. Organisms obtain information from their environment, including light, temperature, moisture, and pressure. For example, if grown in continuous light and unchanging temperature, plants do not develop properly. However, all plants have the capacity to detect at least some environmental factors and alter their developmental of morphogenesis in adaptive ways. So, these factors will act by modulating cellular activity to a particular development into a specific direction, or by cell reprogramming with the restoration of its totipotency characteristic. Growth includes cell division and elongation. During cell division, the symmetry, rate, and plane or orientation of division greatly influence a cell's fate. For example, gut cells, which control gas exchange in plants, typically form through asymmetrical division and by a change in the plane of cell division. However, most plant growth is caused by elongation, which is the permanent enlargement of differentiated cells. Plant cells expand primarily by taking in water. Most of the water is stored in a large central of vacuole. Cell division and cell enlargement determine a plant's shape and direction of growth. However, this process varies among different types of plant cells. The specification of immature plant cells into distinct cell types is called cell differentiation and is also a critical component of plant morphogenesis. Cell differentiation is guided by gene expression change which inactivate or activate protein coding gene. Cell-to-cell communication likely regulates the expression of genes that influence cell differentiation. For example, the root epidermis of Arabidopsis thaliana produce hair cells and hairless epidermal cells. Immature epidermal cells containing one cortical cell differentiate into hairless cells, while those containing two cortical cells develop into root hair cells. This pattern is associated with differential gene expression. Environmental factors such as light, temperature, and the availability of water and nutrients also greatly influence plant morphology. There are three important aspects of morphogenesis to which significant contributions have been made with the aid of plant tissue culture techniques, which are cytodifferentiation, organogenic differentiation, and somatic embryogenesis. In vitro tracheary, element differentiation has long been used to study cytodifferentiation. A variety of experimental systems such as internodes, storage tissues, suspension cultures, single cells, and protoplasts have been used in studies. One of the most significant advances was the optimization of the Zinnia mesophyll cell system by Fukuda and Komamin and Church and Galston. In this case, 50 to 60% of the cells directly without a cell division and fairly synchronously differentiate into tracheary elements within 72 hours. As early as 1939, White had reported controlled differentiation of shoots from tissue cultures of tobacco. In 1957, Scoop and Miller put forth the concept of hormonal control of organ formation, according to which root and shoot differentiation is a function of the auxin-cytokinin ratio. Relatively higher concentration of cytokinin promote shoot formation, and relatively higher auxin concentration favors root formation. This concept proved to of wide applicability. In 1965, it was shown for the first time that full plants can be obtained starting from a single isolated cell. By empirically manipulating the medium and other environmental factors, it has been possible to induce shoot differentiation from explants, callus, cells, or protoplasts of over 1,000 species. It is now well established that plant regeneration can be achieved from all living cells irrespective of their nature of specialization and ploidy level. In 1964, Guha and Maheswari first time reported organogenic differentiation from haploid microspores. The technique of enter and isolated microspore culture to produce androgenic haploid plants is now well established. Even the triploid and the sperm cells have been shown to be totipotent. In 1958, an important contribution was made to the field of experimental morphogenesis when Stewart and Reinert observed the differentiation of embryos in somatic cell cultures of carrot. The development and appearance of these somatic embryos were comparable to the zygotic embryos of this plant. This report attracted the attention of several developmental botanists because until that time, it was believed that embryo formation is the monopoly of the zygote and a few other ovular cells, and embryo development requires a special environment available only inside the female gametophyte or embryo sac. During the last 3 decades, somatic embryogenesis has dominated the research activities in the field of plant morphogenesis not only because of its curiosities, but because of its potential role in basic and applied aspects of plant science. Until recently, plant embryogenesis had been studied intensively to describe morphological and anatomical change that characterize embryo development in different species. Almost nothing was known about the molecular and genetic control of embryogenesis because of the practical problems of inaccessibility to the zygotic embryo, which develop under several cell layers. The progress in the field of somatic embryogenesis during the last 2 decades has been such that in systems such as carrot, gram quantities of practically pure fractions of embryos at different development stage can be obtained. It has therefore now become possible to study genetic and molecular control of morphogenesis during embryogenesis. Using this system, about 21 embryo-specific or embryo-enhanced genes have been cloned from somatic embryos. Investigation related to cellular and molecular aspects of organogenesis and somatic embryogenesis would not only increase our knowledge and understanding of the process involved, but is also expected to enhance our capacity to increase the level of regeneration in presently recalcitrant system. The value of in vitro morphogenesis as a commercially viable procedure for plant propagation and as an essential step in genetic improvement of crops through the modern methods of biotechnology has considerably promoted research in this area. Mass production of genetically identical plants by regeneration from vegetative tissues via organogenesis or somatic embryogenesis is of considerable interest to the horticulturists and foresters. Regeneration via embryogenesis is of particular interest as embryos are bipolar structures bearing both roots and shoot meristems and may allow a certain degree of automation during production and their field planting. Several groups are investigating large-scale production of somatic embryos in bioreactors and their encapsulation to produce artificial seeds to facilitate their mechanized planting in the field. Regeneration of haploid plants from pollen grains have become a popular technique in plant breeding as it's enabled to achieve homozygosity in a single stat, as against several years required to achieve it by traditional method of recurrent selfie. Androgenesis is also serving as a novel source of useful genetic variability. Plant regeneration from protoplast, cell, and tissue as plants is extremely important in harnessing the potential values of somatic hybridization and genetic engineering in crop improvement. Thus, in vitro morphogenesis is of considerable importance in basic and applied areas of growth and differentiation.